Thank you for coming, and I think it was a wise thing to do because I'm really delighted to introduce our keynote speaker this evening, Professor Sonia Sultan from Wellesley University. I first came across Sonia's work, and I'd say this is a confession because I certainly should have known about it before, but I did come across it at a big meeting in London on new directions in evolutionary theory. And I guess really, she gave an extraordinary presentation there on, I think, the transmission of acquired adaptive epigenetic traits. I hope that you would agree with that, was what she was talking about, which I'll say again, the transmission of acquired adaptive epigenetic traits, because it comes out very quickly, but if you slow it down and see what it means that the experiments she did were classically and blasphemously Lamarckian. And again, I don't know whether she's happy with that. Of course, historians will blanch because, of course, Lamarck never said any of the things that we attribute to him. But what the word has come to mean is, I think, what she has shown to happen with her experimental work. If after this, I'm sure you will want, if you haven't already done so, to read her lovely book with OUP from 2015, Organism and Environment, Ecological Development, Niche Construction, Organism and Environment, Ecological Development, Niche Construction and Adaptation, which is a real landmark in the integration of evolution, development, and ecology, which I think is where theoretical biology is going. I think I won't say much more about, she's obviously written lots of fascinating papers and all other kind of good, great achievements that you can find on her website. I think I'll just end on a slightly more personal note. As we were just discussing before the meeting, it's a real sadness that this has to be done in such a way that at the end of it, all that will happen will be a little screen will say the meeting has been closed by the host and we'll all go our separate ways. It would be wonderful to carry on the discussion, and particularly because I remember having a really wonderful discussion with Sonia at one of the evenings of this London meeting. And I was only I'm relieved to hear she remembers it with some pleasure as well, because what I also recall is that I found for us what must surely be the worst Italian restaurant in London. And I guess if it was less well-traveled, she would probably have confirmed all her views about English food, that even an Italian restaurant put into England can provide soggy, overcooked pasta and limp pizzas, and I can't remember what we ate, but I remember it wasn't great. But at any rate, I leave something that I can't do this time, but I hope I will do in the future, which will be share a decent dinner, Italian or otherwise, with Sonia. But for now, a very good second best will be to hear her talk on multi-generation plant plasticity and unscripted developments. Sonia, thank you so much for being part of this meeting.
Well, thank you very much, John, for that kind introduction. And thank you, Özlem, for inviting me to participate in this unusual workshop, which I found extremely stimulating. I have unfortunately missed some of the talks up till now, but I'm in it from now on. My schedule conflicts are over, and I've just, I really hope, even on Zoom, to engage in discussion with this really interesting group and varied group of participants. I think we all probably understand, or the plant biologists understand, that plants are kind of second-class citizens in biology. But apart from the fact that none of us would be breathing without them, I think plants have been a source of some remarkably important and in some cases, as you said, perhaps blasphemous insights about the properties and processes that characterize all living systems. And I'd like to share some of my group's findings about one of those properties, which is plasticity, environmental response. And I think these findings point to some larger questions about the causes of development and about organismic agency. And I'd really like to know what you think of that. So forgive me if I go over familiar ground. I just want to make sure that everyone from varied backgrounds is comfortable with the language I'm going to use and so on. So, sorry, having a bit of a problem here. So, as you know, in a very simplified view of development, an organism's DNA sequence is understood to provide specific instructions for producing that organism's traits. And according to that view, an organism's genotype determines the individual's, use that term advisedly, the organism's traits and its behavior, its phenotype or phenome. In other words, in this view, the genotype is understood to specify a single determinate phenotypic outcome, perhaps with some tiny environmental modulation. And the goal of developmental studies is to reveal this internal, self-contained genetic information. So that is done and has been done for 100 years by raising genetically different individuals in a single so-called control environment that is meant to be developmentally neutral so as to permit the expression of their genetic programs. In this kind of a design, since the organisms develop in the same environment, each genotype produces a specific phenotype. And any differences between them in whatever trait is measured must be due to differences in their genes. So the results of this kind of an experiment will confirm the assumption that phenotypes are determined by genotype. Of course, as Özlem emphasized in her introduction yesterday, the development of organisms does not take place in a neutral box, but in a particular environment. So in the case of this Polygonum plant shown here, for instance, that environment includes specific levels of sunlight, of soil moisture, nutrient ions, atmospheric CO2, root symbionts, plant neighbors, and most likely herbivores and pathogens. And such environmental factors influence the developmental process and consequently help shape the organism's traits. And that is in fact why researchers use a control environment to exclude environmental variation that would influence phenotypes. So thanks to quite a lot of recent molecular biology, we now understand in a different way why the environment influences development. I quote here a very conventional-minded paper that came out in Nature in 2014 that made clear that it is universally accepted that there is tremendous plasticity in gene expression as a result of environmental conditions. So genes are differently expressed in different environments because environmental factors participate in gene regulatory pathways. That is, gene expression and therefore phenotypes are not prescripted in the nucleus, but rather environmentally contingent. So a more informative way to study development is to examine the phenotypes that a genotype produces in different environments. That is, to recognize plasticity, not as a special case, but as a fundamental property of development. Plasticity simply refers to the range of phenotypes produced by a given genotype in alternative environments. So studying these repertoires of phenotypic expression is a way to assess the developmental process and its outcomes in environmental context. And that approach has been termed by developmental biologist Scott Gilbert, ecological development, or eco-devo. So we can implement this approach by inverting the experimental design so as to bring environmental variation into the experiment in a deliberate way. So to do this, we can make several replicates of each genotype which can be done really quite easily with many plants by either cloning them or inbreeding them. So here I show genetic replicates of genotype A and then growing them in each of several different environments. So we can do that for various genotypes of interest. And then for each genotype, we measure the phenotypes it produces in each of the alternative environments and plot the data to show the genotype's response pattern to that range of environments, or what's called its norm of reaction for any trait of interest. So we can study in this way the size of body parts, the physiological traits, such as metabolic rate, reproductive output, life history traits, any aspect of the phenotype. And this kind of data make it clear, make it explicit indeed, that the phenotypes produced by a given genotype depends on its environment. So this kind of study reveals the genotype to be seen to be a repertoire of potential responses rather than a blueprint for one more determined phenotypic outcome. So I want to pause here just to note that the idea of investigating individual plasticity is not new, although the molecular rationale for it is quite recent. And plant biologists in particular have always been aware that phenotypes that are expressed depend strongly on the environmental context. And that is, of course, inconsistent with a narrowly gene-based model, which was actually quite a problem and a dilemma for mid-20th century modern synthesis evolutionary biologists. So if we ask how biologists up till now have reconciled this awareness of environmental context dependency with a view of the genotype as a self-contained developmental program, or a pre-written script for the phenotype, they've done so, we've done so, because I include myself, by defining the repertoire of response, that is the norm of reaction, as itself an intrinsic property of the genotype, as articulated by Gerda de Jong, a very prominent plasticity researcher, an environmental response program written in the genes. So viewing environmental response as genetically scripted has allowed biologists to maintain our primary focus on gene sequence as the source of control over development, and thus as the cause of the adaptive variations among organisms that underlie their ecological interactions and the process of evolutionary change. So this conceptual step of bringing the environmental response program under genetic control, that step matters to our understanding and to our empirical practice, because it justifies maintaining a focus on genes per se. So today what I'd like to do is show some plasticity data, starting simple and then moving to some new results to raise what I think is a key question. Are plastic responses indeed genetically predetermined, or do they take place instead through real-time regulatory interactions in the cells and tissues of the organism, such that we can think of the developmental process as unscripted, as taking place actively? Because if that's a more accurate picture of what's going on, then we have to study and think differently about the causes of variation, the sources of developmental input, and developmental agency. So our main study system for this work is a very common species of annual plant, Polygonum persicaria, which has a number of useful features, including the fact that we can clone them or inbreed them without any inbreeding depression. More on that later. So for these experiments, we collect achenes, seed units from plants in natural populations, clone them or intensively inbreed them, and raise replicates of each genotype in greenhouse environments that are controlled but naturalistic, So we do these experiments under natural summer light, in soil mixes that include local soils, in clay pots that permit gas exchange, not plastic. Very weird and old-fashioned. And our experimental treatments for these experiments are designed based on the kind of environmental variation that the species encounters in nature. So, for instance, light intensity varies among and within real plant habitats, and we use shade cloth and green filters to create contrasting light environments in the greenhouse and ask whether Polygonum genotypes express different phenotypes as a result of being in these alternative environments. And as the plants develop in these treatments, we measure their phenotypes, paying special attention to the traits that influence function and hence fitness. So for instance, at contrasting light levels, and much of the data I'll show you today will be about light, when we look at contrasting responses to contrasting light levels, we focus in particular on leaf traits, which are functionally important since leaves are the organs that collect light. So I'm going to start off with some early work, rather simple, and I promise you we'll go from there. Okay, so one simple result is that plants of the same genotype make larger leaves when they're raised at low light. It's something that has been known for certainly since Theophrastus, if not before. So in this case, these are controlled experiments with individual genotypes on this plot. Each line shows replicate plants of the same genotype that were raised in high light and in low light. So each line is one norm of reaction. And the experiment had 10 genotypes to show these response patterns. On these plots, I'm including a statistics box for anyone who's interested, but don't be distracted by it if it's not useful to you. Okay, so all of the genotypes showed a similar pattern of response, which is to increase the size of leaves that they produced in low light. But I want to point out a couple of other things, in particular, that the size of the differences in size produced by a single genotype in the two different environments were greater than the differences between genotypes within an environment. So these are not minor tweaks. This is actually a formative aspect of phenotypic determination. I also want to point out that the particular slopes of these individual norms of reaction, these genotypic norms of reaction, the slopes differ, which makes sense because DNA sequence influences regulatory pathways that use environmental inputs like light. So we would not, in fact, expect different genotypes to have identical norms of reaction for this trait, for any trait. Okay, this is what the leaves actually look like. You can see the leaves that a plant produces in high light are narrow, which is functionally appropriate since they won't lose too much water. These leaves, you can't see this, but these leaves are also thick with multiple layers of light processing structures to utilize intense light. And the genetically identical individuals in low light produce these large, broad leaves in which the leaf tissue is spread out much more thinly. So I'm mentioning that so you understand that not only the size, but also the anatomical structure of the leaves produced in high versus low light are quite different. When plants are given less light, in this experiment, plants were grown at 3 different light levels. And when plants are given less light, they also increase their the proportion of their tissue that they allocate to leaves. So along with making those larger, thinner leaves, all of those responses maximize their ability to collect photons per gram of plant. Likewise, they increase the proportion of their mass that they allocate to root tissue when they're grown in conditions where soil resources are limited, like dry soil or nutrient-poor soil. So I mentioned these allocational shifts because they can be understood as individual or whole plant adjustments rather than modular adjustments. Because I think that question of goal-directedness in individuals as a potential way to define individuality is a really important one for biologists to consider, as discussed this morning by August and Zoe Nahas. So we've done a lot of this kind of work, and plasticity experiments like this have shown that a single plant genotype will produce very different phenotypes depending on the plant's environment with respect to a number of growth traits, physiology, life history. These are not small tweaks to some kind of pre-existing program. These are major developmental differences that result in very different phenotypes. So the result of all of that work and of a lot of work in many other labs on both plants and animals makes very clear something we really knew all along, which is that genotype and environment jointly shape phenotypes, certainly in the plants that I've been studying. So that's quite clear, but the question remains, is the norm of reaction, is the environmental response pattern a property of the genotype? So to try to get at that question, our next step was to study, as John was mentioning, our next step was to study transgenerational plasticity across generations, and ask if an individual's development can be influenced by effects of previous environments, for instance, by the conditions experienced by the individual's parents. So here's where we get into the Lamarckian territory. Lamarckian is misunderstood, at least in the 20th century. Okay, so the first transgenerational eco-devo experiment we did compared the progeny of genetically identical parent plants raised in two environments. In this experiment, we looked at, we tested for an inherited effect of drought stress experienced by parents. So we raised these genetically replicate parent plants in both dry soil and in moist soil. We did this for, I think it was 8 Polygonum genotypes. Then we collected their progeny and raised the progeny in both dry and moist soil, so as to compare within each growth environment the growth of genetically identical progeny that differed only in the environment, the environment experienced by their parent. So we wanted to ask if we could, if drought-stressed parents, for example, altered the development of the root system in their offspring as a very important adaptive response to promote survival if the progeny also experienced the similar kind of drought stress. So I should point out here that these plants self-fertilize in nature. So one of the very nice features of using this plant for these experiments is that we can use a single plant as both the mother and the father. And that simplifies the design a lot. And that's why I will only refer to the parent plants, because in each case there's one parent. Are you with me on the design? Any questions? Okay, I hope that's clear. Okay, so again, we paid attention to the root system because that's the key to plant function in dry soil. So we actually, and when I say we, I mean my students, brave souls, who monitored the development and extension of roots over time in each individual progeny. So the results were quite striking. When they were grown in dry soil, the seedling offspring of drought-stressed parents, here shown in red, extended their roots more deeply, faster than did genetically identical offspring of parents that had been given plenty of water. So the root difference was quite substantial, and it got bigger actually as they developed. And that resulted in much longer roots, 25% longer roots at about day, harvest was about day 25, and 36% greater biomass in these progeny of drought-stressed parents compared to the progeny of plants, parents that have been grown in moist soil. And again, the only difference between these two groups is the parent plant's environment. They're genetically identical. So when they were growing in dry soil, the two groups of progeny showed this very strong difference. But this inherited effect of drought stress was not expressed when the two sets of progeny were grown in moist soil. So that indicated something really surprising, which is that individual seedlings seem to be integrating past and present environmental signals in a way that led to a particular developmental outcome. We expanded this study over another generation of inheritance because we wanted to know whether that kind of inherited environmental effect on development could persist for longer than one generation, because that, that opens up a whole lot of other questions. So to expand the experiment, we produced isogenic seedlings, again, seedlings from each inbred genotype, of all 4 possible combinations of grandparent and parent environment. So we grew grandparent plants of each genotype in dry and moist soil, raised their progeny in dry and moist soil. Those are the parent generation. And then we ended up with 4 sets of grandkid seedlings, each of which differed, sets which differed only in the 2 previous generations of environmental conditions, but were genetically identical. So we have these, for each genotype, we have 4 sets of seedlings. DDs had a drought-stressed grandparent and a drought-stressed parent. MDs had a moist-grown grandparent and a drought-stressed parent, and so on. And then, because this experiment was already quite big, we grew all of these seedling progeny only in dry soil. So we only compared their development in dry soil. And again, we monitored root development very carefully. So that was interesting too. It just goes to show in biology, you can only get an answer if you ask the question. And we've been asking some questions that I think just hadn't been asked. Okay, so this plot shows the deepest root produced, so the root depth achieved at a certain time by plants of the 4 combination treatments of drought and moist soil in the 2 previous generations. And what we found is that seedling root extension, again, all of these plants were growing in dry soil. Root extension was influenced by both previous generations of the environment, both grandparent and parental drought stress, including this, which shows a persistent effect of drought stress in the grandparent generation, even over an intervening generation of moist grown parents. In other words, plants that experience seedling grandkids whose grandparents had been drought stressed, but not their parents, grew longer roots than those with no history of drought stress in either the parent or the grandparent generation. So the persistence of the grandparent effect across the parent generation indicates an epigenetic mechanism for this effect rather than a direct maternal cytoplasmically transmitted effect from in building the seed. And later work by the graduate student who led this project, Jacob Herman, showed that this effect, the inherited effect of drought stress on seedling root development, is indeed mediated by DNA methylation. And I should also mention that these seedlings, those with 2 prior generations of drought stress, also survive significantly more in dry soil than those with no prior history of drought stress within the same genotype again. So these 2 previous generations of environmental conditions are important to fitness. And one of the really big questions about these epigenetic environmentally induced epigenetic effects is how many generations they can persist. We know in some systems it's very short-lived, maybe 1 or 2 generations. In other systems, it can be quite a long time. In C. elegans, a nematode, there's been persistence for 40 generations. We tend not to know the answer to that question because we don't tend to do our transgenerational environmental experiments for that many generations because life is short. So we may know the minimum number of generations that in effect persist, but not the maximum number. Okay, I want to show you one more result, which I'll return to later, which is, this is a different trait. This is germination day, the same for set of grandchildren, seedlings. And what I want to point to here is an interaction effect between the genotype, the grandparent environment, whether it was dry or moist, and the parent environment. Because this suggests that the environmental effect of the grandparent stress and of parental stress interact with each other, which means the particular sequence of those environments matters. And that they're not inherited as fixed effects. The effect of the grandparent drought depends on the parent drought. Okay. So here's where we are based on all of that both genotype and environment, current and ancestral environment or environments, we don't know, jointly shape the adaptive phenotypes organisms express in ways that matter, and important developmental traits relating to fitness, such as root development for seedlings in dry soil. Okay, does that insight change anything? I think it does. Now, I should also point out that this point, the point about the effects of previous environments on phenotypic expression, has also been found by many other labs studying, again, both plants and animals. Okay, so to try to understand what this might mean for us, I want to show you some more recent work on transgenerational effects of parental sun and shade on offspring development. So for this, I'll again focus on leaf traits. And I want to use this slide to make one more, what I think is a very important point. So in this slide, each box shows the size of leaves produced by seedlings of a single genotype grown in either sun or shade for 5 different Polygonum genotypes. But, and this is again, leaf size. But in this case, each genotype's plastic responses to these 2 growth conditions was very different, depending on whether its parent had grown in shade or in sun. So here, we're looking at 2 norms of reaction for each genotype rather than 1. And beyond that, how much those 2 norms of reaction differ from each other, that is, the effect of the parental environment varies from one genotype to another. So in this genotype, there's a very subtle effect, if any, might not even be a significant effect. In this genotype, there's quite a big effect for progeny developing in both sun and shade. In this genotype, there's a small effect for progeny developing in sun, but a very big difference in the leaves they make in shade as a result of the environment of the parent. And that result for me was a kind of a jumping off point, because it means that the inherited effects of previous environments participate in determining norms of reaction. And that suggests that the developmental responses of individuals to their own environments are not scripted by the DNA sequence. Those are not genotypic properties. Instead, that response to the alternative environments that the individual itself faces arise from something that's happening in real time, from the regulatory interactions that take place in cells and tissues that integrate environmental inputs, present and past, with genetic information. So that's the result that we're following up on now with our current project. And for the last few minutes, I'll show you a few of the initial results from that project, which again uses sun and shade as the alternative environments. But in this case, we're using a larger genetic sample. We call this study the G/E² study, because what we want to do is to track the interactive effects of genotype, parent environment, and current environment on phenotypes across the life cycle, and to get a more fine-grained picture of that interplay of developmental influences. We're using a sample of 12 genotypes. And we're also examining environmental effects on gene expression more directly by looking at the transcriptome. So there's an RNA-seq parallel, which I can't show you today, to our results. So this is an enormous experiment carried out under very difficult conditions last summer by an extraordinary group of students and postdocs who I'm very grateful to. I will show you their names in a bit. Okay, so for this study, first we looked at the impact of parental and offspring light, sun versus shade, on seedling emergence, which is of course a very important life history trait for plants in the real world. The vast majority of plant mortality takes place very early in life. So this trait, for example, can result in a slight advantage over near neighbors and can be a life or death trait, like many of these early seedling traits that we're studying. This plot shows the percent of emergence as of day 3. So a higher position on this plot means faster emergence from the soil. So what we found is that seedlings emerged slightly faster in sun than in shade, and very much faster if their parents had been shaded. So these lines are the means across all 12 genotypes in the study. And they showed this overall effect shows a very profound effect of parental environment on emergence timing. But we actually track timing in detail. This is across time, days since sowing, in each genotype. So this plot shows a box for each of the 12 genotypes in the study, and there's 4 lines in each box, because for each genotype we had 4 environmental combinations of parent and current sun or shade. So for each genotype, we can look at the response trajectory for emergence time. Offspring of shaded parents are in this blue color when germinating in the sun, dotted or when germinating in the shade. And offspring of full sun parents are in this orange color, again, when germinating in the sun versus when germinating in the shade. So, the point that I want to make using this very complex looking slide is simply that the 4 parent offspring combination environments played out differently from genotype to genotype. That is, the 12 genotypes responded differently to combinations of parental and offspring light conditions. We used imaging to study the canopy area produced by each plant across its life cycle. Canopy area is, of course, a key functional trait, especially in shade, and is especially important very early in development, again, for developing, for getting a good start and competitive performance with neighbors and so on. So this is a really nice technique where you take a photograph and desaturate it and then binarize it using a thresholding algorithm so that you turn it into black and white, and then you can digitize it and do actually quantify canopy area. It's a complex process to do this analysis, and we're working on it, but today at least I can show you the results for canopy area of individual seedlings at this developmental stage, very early in life, around day 16. Okay, so I made the slide yesterday. thanks to one of my postdocs, Michelle DeGuillo, for putting it together very quickly for me. Here's what we saw. So again, each box is one genotype growing in sun or in shade, and the parent environments are the two different colors for each genotype. And what we found is that in all of the genotypes, again, as young seedlings at this very early time point, the offspring of shaded parents, shown in blue, produce larger canopies. This is the canopy. Than genetically identical offspring of full sun parents. That difference was especially pronounced for most genotypes when growing in shade. But again, this effect and the combination of parent and offspring condition effects varied among the genotypes. Okay, this is the very last bit of data I'm going to show you, which is data for seedling height, which we measured in seedlings and then in young juveniles to start looking at developmental interactions among genotype, current, and previous environment, to look at those interactions over time. And actually, that's been the real beauty of looking at this data, is to watch the developmental process unfold and watch how these influences come into that process in different ways across time. So here's the last data slide. I want to take just a minute to unpack this because I think it has a really interesting message. Okay, this plot again shows each of the 12 genotypes, but here you're looking at these at the genotype at two developmental time points. That very early time point that you just were looking at photos of, and slightly later when it's kind of a young juvenile. And we're looking at plant height expressed in shade and sun, and in respect to these two parent treatments at two different time points. So I'll show you a few examples to try to convince you that these genotypes differently integrated parental and current environmental influences over time. So what do I mean by that? Well, for instance, in this genotype, there's a slightly larger effect of parental shade versus sun on day 17 than there is on day 28, but only on the height of seedlings growing in shade. On the other hand, in this genotype, there's no effect of the parental environment on height at all on day 17. There is an effect of the current environment, but there's a substantial effect of the parent environment later in development, a bit later, day 28, but only on height in sun. This genotype also showed a bigger effect of the parental environment relative to the current environment later rather than earlier, but in this case, that parental effect was expressed only in shade, only in seedlings growing in shade. And in this genotype, there's no effect of the parental environment. There is a strong effect of the current environment, no effect of the parental environment at either time point in either developmental environment. In other words, the process of developmental integration, the integration of previous and current environmental influences with genetic factors differs, or let's say the integration of previous and current environmental influences differs from one genotype to another. And because it differs, the differences are genetically inflected, the process itself theoretically can evolve. In other words, it can be seen as an aspect of genetic diversity. But if you think about that, it could only evolve through a selective process that's mediated by these very high order environmental interactions. In other words, a very noisy, very buffered selective process, rather than the kind of direct signal from genotype to phenotype that underlies our way of thinking about the selective process, which is a very clean targeting of better outcomes. Okay, so that's been a lot to look at. What does all that plasticity data mean? I'm not certain. Okay, there's a lot to think about. I think it means this, that the organism's developmental response to its environment is not scripted in its genotype, but instead that response gives way to produces a phenotype that takes shape through real-time regulatory processes that integrate past and present environmental inputs in genotype-inflected ways. There is certainly a role of genotype. It is not a deterministic role. So if that's a more accurate picture, it points to a more distributed model of phenotypic causation that includes environmental inputs across generations, which is a much harder and more interesting thing to study. And in fact, since environmental influences are inherited, these influences are just as internal, just as inborn to the organism as its genotype. And that means, in biological terms, that the genotypes that we study are actually what Evelyn Fox Keller has called genotype environment entanglements. Potentially, entanglements accrued over several generations, maybe even multiple generations of environmental effects, which can possibly interact with each other, as in that one case I showed you, where the effective grandparent environment interacts with the effect of the parent environment. That's a very complex picture of causation. And it raises all kinds of operational and interpretive problems for biologists, because after all, our goal, our prime directive for decades has been untangling environmental influences from the genotype. And if environmental influences are built in to what we study as genotypes, we need to reconsider how we study them. And we also need to reconsider the goal. What would it even mean to peel off all of the environmental influences? Okay. So that's where the data are. Because this group is such an interesting one and has raised so many fundamental questions about plants, I wanted to just raise one, which is the question of whether plant responses are cognitive. I don't know. I think certainly there are perception and memory and action at work. So those are the three criteria that John gave us yesterday. I'm not sure I would use that word, but if these responses implement goal directedness, directedness toward biological goals, then there is a form of agency in the responses of organisms to their environments that our very single-minded focus on genetic control as biologists has really missed. And we might find it, or find it better, by studying how development really works, which means bringing the environment in. Okay, thank you very much for your attention. And I also want to thank, with deep gratitude, the many members of my lab, past whose work I showed today, and also in this color, the G by E squared team, which labored just valiantly under these difficult conditions last summer to move this work forward. Okay, thank you.